Hello, my name is Pam Ronald. I'm from the University of California, Davis. Today I'm going to talk about plant biology research, which is critical if we're going to address one of the greatest challenges of our time, how to feed the growing population without further destroying the environment. We know that the population is increasing from a current 7 billion to possibly 9 or 10 billion at the end of the century. And we can't feed more people by plowing more land because most of the arable land has already been farmed. This is a picture of a hillside in Ecuador, and you can see farmers are farming increasingly marginal lands, such as these very steep hillsides. Another issue is water. Already 70% of fresh water is used for agriculture, and demands for water are increasing over the next 50 years. Plants are susceptible to diseases, as are humans, and this is an image of a tobacco plant that's infected with a very serious pathogen that is also a related pathogen that was responsible for the Irish potato famine, which in 1850 contributed to the deaths of 1 million people and caused another million people to emigrate. This is a time-lapse photo over a period of 4 days. So you can see how devastating plant diseases can be. We can control diseases somewhat by using pesticides, but these pesticides, some of them are toxic and many of them cannot be used safely. So this is a potato farmer in Peru, and you can see that he is spraying his potato farm, but he has no protective gloves, he has no mask, and as he poses for the camera, he's spraying his boot. So this is one of the reasons, this lack of safety procedures, that 300,000 deaths occur every year due to pesticide poisonings, and most of these deaths occur in the less developed countries. In addition to pests and disease, climate change is predicted to reduce agricultural production in the future. The Intergovernmental Panel on Climate Change predicts that agricultural yields will be reduced 2% per decade in the coming years, and that is at a time when we need to increase production instead of reduce production. Environmental effects of climate change include drought and heat, but also flooding. And we're going to see more and more floods in areas where we don't want to have floods. So this is an example of boys walking through their family's rice field. These rice fields can be flooded periodically. And although rice does grow in water, if rice is completely submerged, most rice varieties will die within 3 days of flooding. And this is particularly important in areas such as Bangladesh, where people get 2/3 of their total calories from rice. So today I want to tell you the story of flood-tolerant rice, which is a collaborative project over the last 10 years. 25% of the world's rice is grown in flood-prone areas, shown here in this red circle, including eastern India and Bangladesh. Now, in this area, just in Bangladesh and India alone, 4 million tons of rice, enough to feed 30 million people, is lost every year to floods. About 50 years ago, there was an old rice variety that was discovered that had an incredible property. It could withstand 2 weeks of submergence. That is, that after the flood subsided, this rice variety would survive the flood. Now, breeders using conventional methods tried to introduce this trait, which is encoded by a gene or genes, into varieties that were locally adapted and favored by farmers. However, the new varieties did not carry the characteristics that the farmers were looking for. So several years ago, a collaborator of mine, David McKill, and I decided to try to isolate the gene encoding this important trait. We were able to isolate a gene called sub1A using a technique called positional cloning that confers submergence tolerance to rice varieties that normally would die in a flood. And you can see an example of this here. On your left is the control rice strain before submergence, and on the right are 2 genetically engineered varieties carrying the single gene called sub1A. After 16 days of submergence, you can see the control is the leaves are drooping, they're yellowish, chlorotic, and elongated. And the two transgenic varieties look pretty good. Now, this is what the plants look after 7 days of recovery. So the flood is removed, and the plants are given a chance to recover. The control plant on the left has died, whereas the two genetically engineered plants carrying the sub-1a transcription factor are completely healthy. Now, my collaborators, Julia Bailey-Sayers, Takeshi Fukao, and Haruka Malwalia, has started to look at the molecular mechanism. And the way sub-1a works is that it sort of activates a hold-your-breath strategy. And this allows the plant to conserve its energy until after the flood subsides. So for example, submergence is known to increase local concentrations of ethylene. Now ethylene then upregulates the sub-1a transcription factor, which then induces ethanol fermentation genes. Sub-1a also suppresses alpha expansins, which are important in leaf elongation. And sub-1a also suppresses another related transcription factor we identified called sub-1c. Normally, in the absence of sub-1a, submergence would induce ethylene, which would elevate gibberellic acid, and that would turn on sub-1c. And sub-1c is a positive activator of cell elongation and carbohydrate breakdown. So sub-1a is a key regulator of this process and essentially shuts down cell elongation and carbohydrate breakdown. And Haruka Malwalia at my lab has also been able to show that this upregulation of sub-1a also upregulates genes that are important for biosynthesis of the hormone brassinosteroid. And brassinosteroid then activates some very important genes, including SLR1 and GA2 oxidase 7. Now, GA2 oxidase 7 actually contributes to the breakdown of gibberellic acid, and SLR1 suppresses gibberellic acid-mediated processes. So this is another level of control of sub-1a controlling cell elongation and carbohydrate breakdown. Now, if we had isolated this gene and done some interesting biology, that would have been great for basic plant biology. However, the goal of this project always was to try to develop a better rice variety that can be used by farmers in Bangladesh and India and other areas of the world. And this is my collaborator, David McKill, who was at UC Davis and then at the International Rice Research Institute. And he led a team, including Abdel Ismail and many other researchers, to introduce sub-1a into various rice varieties that are favored by farmers. And in his approach, he used what's called marker-assisted breeding. So in the next slide, I will show you the effect of the introduction of the sub-1a gene using marker-assisted breeding. So this is a time-lapse sequence that you're going to look at. So it's a 4-month time-lapse that was filmed at the International Rice Research Institute. And you could see the effect of the sub-1 gene on a variety of rice called IR64 that was developed by David McGill, Abdel Ismail, and their team. So what you see here on the left is the new variety that was developed carrying the sub-1 gene, and on the right is the control. And here's an artificial flood that was placed for 17 days. And what's interesting is that you could see immediately that the IR64 carrying the sub-1 gene is highly tolerant of flooding, whereas the control has very poor yield. And I really like this video because it shows the power of genetics, the power of a single gene to transform the yield of a very important staple food crop. Dave and his team led 4 years of field trials, and our team was fortunate to visit the farmers in their field. And over the last 4 years, when these tests were done, there was a lot of flooding every year. So these are conditions that were not induced in a field crop, but were observed in farmers' fields. So you could see the farmers on the left that planted the conventional variety Swarna, and on the right is the new variety carrying the sub1 gene. What the farmers found is a 3 to 5-fold yield increase in these new varieties. We also had an opportunity to speak with many of the farmers and their family, and this is a woman explaining what she thought of the new rice variety. So one advantage of this project was that it was a fantastic collaboration, not only between scientists in my laboratory at UC Davis, but also scientists at the International Rice Research Institute and breeders at the Bangladesh Rice Research Institute, and also other rice research institutes in Bangladesh and India. I want to now turn to another critical issue within crop production, which is disease. And one of the goals of plant biologists for many years, over 100 years, has been to to enhance the resistance of plants to disease. Plants and animals are infected by many different kinds of microbes. So for example, here is a rice leaf that is infected with a gram-negative bacteria called Xanthomonas oryzae, pathovar oryzae. You can see the bacteria, which has a yellow pigment, oozing out of the rice leaf. This is a dead fly that's infected with a fungal pathogen, Aspergillus fumigatus. And here is a baby that's infected with a devastating disease that when it becomes septic, it can become invasive. We now know, after 15 years of research, that the plants and animals carry similar genes to resist these devastating diseases. First, some history. In 1845, late blight destroyed the Irish potato crop. This serious disease caused the death of 1 million Irish farmers and their families, and contributed to a mass exodus to the United States of another 1 million people. At the time, it was not known what caused this disease. It was thought to be an act of God. However, this disease triggered the first serious investigation of diseases of plants and animals. In 1853, Antoine de Berry demonstrated for the first time that microbes can cause disease. He was studying rust and smut fungi on cereals. Louis Pasteur, who knew of Antoine de Berry's work, then showed that bacterial contamination caused what he called diseases of beer and wine. And it wasn't until 1876 that Robert Koch showed for the first time that animals also are infected by microbes, and these microbes can cause disease. In 1905, Roland Biffen used this new knowledge on the genetic basis of disease to develop the first varieties of plants that were resistant. And he was studying cereals. He was able to show that he could cross a resistant wheat variety with a susceptible wheat variety, and that resistance would be inherited to the next generation. Today, virtually everything we eat carries genes for resistance. These resistance genes are critical to economic productivity and are the most safe and economically effective method of controlling disease. So what are the genes that confer resistance to infection? For the first 100 years after Roland Biffen's discovery, breeders were introducing these genes for resistance without knowledge of really what they were or how they functioned. And when I was a graduate student in the 1990s, this was a really very important question to really understand the genetic basis of disease resistance. And my research has primarily been on the bacterial blight of rice. You can see the yellow pigment that this gram-negative bacteria forms in culture. And this is a disease that occurs in Western Africa and throughout South and Southeast Asia. So we began this work with the help of Gurdeep Kush, who is now at the University of California, Davis, but he spent most of his career at the International Rice Research Institute. He's considered one of the fathers of Green Revolution rice, and he was awarded the World Food Prize for his work in enhancing rice production for farmers in very poor and less developed countries. Now, in collaboration with his colleagues, he discovered a very unusual wild species of rice called Oryzae longistaminata. Now, this wild species had an incredible property, which it conferred broad spectrum resistance to virtually all strains of Xanthomonas that were discovered in the field. So I was very inspired by this work and decided to try to isolate the gene. We were successful, again, using a positional cloning strategy. We isolated what's called the XA21 gene. And you can see on your left the control rice variety called type A309, infected with the bacterial blight pathogen. And what we do is simply dip scissors in a bacterial suspension, clip off the top of the leaf, and wait 2 weeks and follow the lesion development. And you can see on the right is the genetically engineered rice carrying this gene XA21 from a wild rice species. And this rice now is completely resistant to disease. So what is this gene? Well, it turns out that it's a receptor kinase. So it carries a leucine-rich repeat domain on the outside of the cell and a transmembrane domain and an intracellular kinase domain. So it's a receptor kinase that senses the microbe outside the cell and then triggers a defense response inside the cell. And this story became very interesting because in 1996, the toll receptor from Drosophila was isolated, which had a very similar structure to XA21. It had a leucine-rich repeat in the extracellular domain, a transmembrane domain. It had a small intracellular domain called a toll interleukin receptor domain, and it associated with a kinase called Pele. And I should mention that this toll interleukin receptor domain is very similar to a domain that's present on our resistance gene in tobacco called the N gene, which you can see here. It was discovered in 1994 in Barbara Baker's lab. There's a nucleotide binding site, a leucine-rich repeat domain, and a toll interleukin receptor domain. In 1998, TLR4 from mice was isolated by Bruce Beutler's lab. It had an identical structure to the toll receptor. And you can see that in addition to the leucine-rich repeat domain, TLR4 also associates with an intracellular kinase called RIP1. In the year 2000, Arabidopsis FLS2 was isolated. This is a model plant used for genetic studies in plant biology, and it had an identical structure to XA21. And in the year 2001, the TLR5 receptor was isolated. This is another mammalian receptor, which not only has the same structure as TLR4 and toll, but it recognizes a very similar microbial molecule as FLS2. They both recognize a small peptide called flagellin. So at this point, we knew that plant and animals use very similar strategies for defending themselves against disease. And another important point is that although kinases are abundant in both plant and animal genomes, there is about 500 in humans, about 1,500 in rice. There is a small subclass of these kinases called non-arginine aspartate kinases that make up about 20% of the kinome in humans and 25% in rice and Arabidopsis. And it turns out that this class of kinases, the non-RD kinases, are critical for activation of the immune response via these leucine-rich repeat type receptors. So we know that immune receptors in plants and animals are remarkably similar. And the discovery of these receptors revealed a previously unknown mechanism of immunity in both plants and animals. And it was really a very exciting time for both plant biologists and animal biologists to see the merging of the fields of immunity. So I want to, that was a little history, and bring you up to date to some of the types of things we're doing to understand the XA21-mediated immune response. Now, we've used standard molecular techniques such as a yeast-2 hybrid system, which looks at protein-protein interaction. We've done transcriptomics, looking at changes in gene expression. We can look in Arabidopsis or animals and predict if similar proteins transduce the rice defense response, and we're carrying out several genetic screens to identify other genes that are critical for this response. As you can imagine, these studies have led to a massive accumulation of data, and we still have many gaps in our knowledge. So we needed help. We turned to computational biologists, Edward Marcotte, and at the University of Texas, Austin, and In-Suk Lee, who is now at Yonsei University. They have been developing very interesting computational tools. And they were able to develop a tool called RiceNet, which collected 50 million data points from 23 types of data sets and 5 different species. And this allows us to identify genes that function in the same biological pathway. From this work, they were able to link about 45% of the predicted 41,000 rice genes. Now, what can you do with this type of model? Well, what you can do is you can query it with genes that you know are important for a response and try to identify other genes that function in the same pathway. So that's what we did. We queried RiceNet with genes that we knew were important for the XA21-mediated response. And those are the genes shown in red. So these are genes that we know genetically are critical for the response. And the balls in green represent new genes that are predicted also to be involved in this pathway. This led to identification of hundreds of unknown genes, and we can't analyze all of them. So we used a prioritization step to try to figure out which ones we're most interested in. So what we did, so on the left in green and white, are genes that we had identified over the years that were important for the XA21-mediated response. And we decided that some of those network-predicted genes, if they were truly important, they might interact physically with some of our other genes and proteins that we had identified. So we carried out a pairwise protein-protein interaction test, and we identified a set of about 12 genes, shown here in yellow, that encode proteins that interact with some of our other very important proteins in the pathway. And then we picked 5 of these to analyze genetically, because through genetic analysis, by knocking out expression or overexpressing the gene, you can try to understand if alteration in gene expression affects the defense response. And we've identified 3 new regulators of XA21-mediated immunity by this approach. And so they were first predicted computationally by RiceNet, and they were prioritized using a protein interaction assay, and then they were validated in whole plants using an XOO infection assay. Now, these genes we're just beginning to characterize them. The first one is called ROX1, which is a thymine pyrophosphokinase, which, interestingly enough, has also been implicated in some types of human brain disease. So studies in rice can lead to the identification of genes that may also be important for controlling the disease response in humans. Similarly, ROX2, which is a NOL1 NOP2 domain family protein, has been implicated in a childhood disease called the Williams-Burren syndrome. And ROX3 is a NUDC nuclear movement protein, and orthologs in animals have been shown to be involved in tumor proliferation. So where are we with XA21-mediated immunity? We know a lot more than we knew 15 years ago, but there's still a lot to be discovered. So we believe that XA21 is synthesized in the endoplasmic reticulum with a chaperone called BIP3. And we've recently shown that the XA21 receptor kinase associates in vivo with another co-receptor kinase called SIRK2, which is critical for function of this XA21-mediated response. There's another protein called XA21 binding protein 24, or XB24, which encodes an ATPase that binds with XA21 in vivo and promotes autophosphorylation of the XA21 kinase domain. Now, autophosphorylation on these particular amino acid keeps XA21 in a resting state. Upon pathogen infection, the XB24 XA21 complex releases, and we then can activate the immune response via several other proteins such as XB3 and a new protein called a receptor-like cytoplasmic kinase that my collaborators, Xue Chen and his lab, have recently discovered. And this is thought to trigger a MAP kinase cascade. We've also identified a group of transcription factors called Werke transcription factors that are similar to the mammalian REL-related transcription factors that are critical for regulating this response. Eventually leading to the immune response, and this whole process can be negatively regulated by a phosphatase called XB15. So one of the important projects in my lab is to try to identify the microbial molecule that activates XA21-mediated immunity. So in other words, what is the microbial molecule detected by the Rice XA21 receptor? And for this, we've started to work on the bacterial, to carry out bacterial genetics. And the goal is to identify Xanthomonas genes that are required for activation of XA21-mediated immunity. And we call these genes Rax genes. And now I'll briefly tell you about two groups of Rax genes that we've identified. Rax A, B, and C are genes predicted to control type 1 secretion, and the genes Rax ST, Rax P, and Q are genes predicted to control sulfation. First of all, Rax A and B are in a small operon, and they encode predicted components of a type 1 secretion system. That is, they work together with another protein to form a pore where molecules are secreted from inside the bacterial cell to outside the bacterial cell. And RaxB encodes a particular type of ATP binding cassette transporter that has a C39 peptidase domain on the N-terminus, which is predicted to cleave a leader sequence from a small peptide, and then after cleavage of that sequence, it's secreted outside the cell. Now, these Rax A and B orthologs in other species are predicted to function with a third protein called an outer membrane protein. And through orthology search with E. coli, we were able to identify such a protein in Xanthomonas. When we make mutations in either Rax A, B, or C, the bacteria are impaired in their ability to activate XA21-mediated immunity. We've also identified genes predicted to control sulfation, and those are called Rax PQ and Rax ST. And these genes work together to add a sulfur residue to a substrate. So, for example, Rax P and Q work together to encode an ATP sulfurylase, and they convert sulfate to adenosine phosphosulfate, and then Rax Q encodes an APS kinase, which converts APS to phosphoadenosine phosphosulfate. Now, this molecule, PAPS, is critical as a substrate for the action of the sulfyltransferase Rax ST, which can use the PAPS to catalyze sulfation of a target substrate. So our model now is that a secreted sulfated peptide activates XA21-mediated immunity. And that's shown here again. Rax P and Q work together to form phosphoadenosine phosphosulfate. And that sulfate is utilized by the Rax ST sulfyltransferase to transfer sulfate to a predicted tyrosine residue on an unknown substrate that we're now trying to identify. And that sulfated substrate is secreted outside the bacterial cell via the Rax ABC type 1 secretion system. And we're also very interested in this because similar systems have been shown to be very critical in agriculture. So for example, Cynorhizobium meliloti is a gram-negative bacteria, and it sulfates what's called the NOD factor, which is a lipochitooligosaccharide, which is required for specific recognition by its host, alfalfa. And this process establishes the ability of the microbe to fix atmospheric nitrogen, which is very critical for agricultural production. We also know that in the HIV virus, for attachment to human cells, it requires the CCR5 co-receptor, and this co-receptor must be sulfated. That it's a key recognition of the HIV envelope glycoprotein. And humans that don't have CCR5 are completely resistant to HIV. And finally, we know in carrots that a very small sulfated peptide called phytosulfokine binds to carrot receptor kinase, which controls cellular dedifferentiation and proliferation. So we know then that sulfated molecules generally are directed outside the cell where they modulate cell-cell interactions. So a few years ago, my laboratory established a collaboration with Lina and Jendra Tripathi at the International Institute of Tropical Agriculture in Kenya. And we were very interested in trying to develop methods to control disease of banana. 100 million people in East Africa rely on banana as a staple food source. And one of the devastating diseases of banana is called banana Xanthomonas wilt. And you could see here a cross-section of a banana and the bacteria oozing out of the stem. This bacterial infection attacks all the banana varieties. There's no known genetic resistance, and there is no way to control the disease. Once the plant is infected, it leads to complete crop loss. The methods to prevent infection are very laborious and crude. Farmers need to clip off the buds on the banana plants and use very clean tools. However, once the infection progresses, the farmer has no choice but to either burn the entire field or destroy the crop using an herbicide and start again. So scientists around the world are trying to develop methods to control this disease. Now, we discovered recently that the bacterial genes in the rice pathogen that we were studying are very similar to genes that are present in the banana pathogen called Xanthomonas campestris pathovar mucicerum. Now, this similarity of genes suggested to us that the banana pathogen may also produce a molecule that's critical for activating the immune response in the presence of the XA21 rice receptor. So we carried out a simple experiment. Lina and Jendra introduced the rice XA21 gene into banana. And they then took those bananas and inoculated them with the bacteria that causes banana Xanthomonas wilt disease. And they've recently shown that bananas, this is the result of their experiments. On the left, you can see a banana that's completely susceptible to the disease. And on the right are their genetically engineered bananas. These are two independently transformed lines, and they've generated 6 lines that are completely resistant to disease. So we're very excited about these results, that a simple experiment putting a rice gene in a banana can have such a dramatic effect. Now, if these experiments can be replicated in the field, this will be, we believe, a tremendous advance for farmers in Eastern Africa. Now, putting a rice gene in a banana has not been done before, and many consumers question whether tampering with nature in this way can create some types of unintended consequences. And so this question is something that national academies, scientific academies around the world have looked at over the last 20 years. And over these 20 years, we've accumulated a lot of scientific evidence and data. So I would like to just summarize the scientific consensus, and this is based on 2 billion acres of genetically engineered crops and 20 years of these crops planted. And the consensus is that the crops currently on the market are safe to eat, that there are clear environmental benefits, and that's mainly relying on the studies of cotton, BT cotton, which has massively reduced sprayed insecticides, and their predicted health benefits. And that is because of golden rice, which we can talk about later. That's expected to be released later this year or next year, which is enriched for vitamin A. all processes of genetic alteration, whether it's a conventional approach or more modern approaches of marker-assisted breeding and genetic engineering, present some risk of unintended consequences. So there's not a situation where you have no risks, but those risks must be weighed with the benefits. We know that over-reliance on a single herbicide will foster herbicide resistance in weeds, so there's big efforts around the globe to try to develop integrated management for weed control. We know that each genetically engineered crop must be evaluated on a case-by-case basis. There's no scientific reason to group all genetically engineered crops together. Every crop is different, every trait is different. So these are the conclusion of the European Food Safety Authority, which is the highest authority for food safety in Europe, as well as the World Health Organization and the American Association for the Advancement of Science, as well as every other scientific organization that has examined genetically engineered crops. And I'll show you some of these organizations here. So I'd like to conclude with a subject that's important to my husband, Raul Adamczak, and myself, which is how is the best way to enhance sustainable agriculture. So he's been an organic farmer for 30 years, and here he is at the University of California campus, where he runs an organic farm and a teaching program for students. And many people think that a geneticist and an organic farmer rest on opposite ends of an agricultural spectrum. But we don't, because we have the same goal, which is an ecologically based food system. Still, many of our friends and family have asked us if genetically engineered crops are safe to eat and whether organic farming can feed the world. So in response to those questions, we wrote a book called Tomorrow's Table: Organic Farming, Genetics, and the Future of Food. And what we tried to do in this book is distinguish between fact and fiction on the discussion on crop genetic engineering, and also bring readers into the lives of what an organic farmer does every day, as well as a geneticist. And we believe that it's important to advance sustainable agriculture using the most appropriate technologies. And sustainable agriculture rests on three pillars: the social, economic, and environmental consequences of a particular crop production system. Rather than focusing on how a seed was developed, we need to ask important questions as to who benefits from a particular technology. What are the economic impacts on a farmer's ability to sell their produce and produce their food and feed their families and to enhance local food security? And importantly, what are the environmental effects of each new technology or farming practice? We need to reduce the amount of toxic inputs into the environment, and that includes some insecticides and some herbicides, as well as conserving land and water and fostering soil fertility. Thank you, and I'd like to acknowledge several of my sponsors, as well as a really dedicated and wonderful team and collaborators. There are many people in my laboratory that worked on isolation of the XA21 gene and isolation of the sub-1 transcription factor. That work was David McKill and his group were very critical for this work. Yonsei University, as well as University of Texas, Austin, my collaborators Insuk Lee and Edward Marcotte and their team were critical in developing the computational predictions. And my collaborators at UC Riverside had wonderful contributions to understanding the mechanism of sub-1 action. And my recent collaborators, Jendra and Lena Tripathi at the International Institute of Tropical Agriculture, have engineered banana to carry the rice XA21 resistance gene. So thank you very much for your time.